technical difficulties always seem to poke their head up after lunch. So I will just quickly ask everyone's patience while I thank, of course, Kristofer for being here and point out that Vince will be moderating today. There is a Q&A mechanism available for all the participants. It's at the bottom of your screen there. If you open it up, you'll see the opportunity to post questions and upvote them if you wish. Please use that during Kristofer's talk so that Vince can then read these questions at the conclusion of the talk for Kristofer to answer. And now I'll get out of the way and let's use what's left of the time to enjoy the talk. Thanks.
Mark, you need to enable me to share screen.
Absolutely, thank you. You have the power.
Perfect.
All right, so let's get started. Let me just do this. Introduce Kristofer Bouchard. He is a computational biologist and currently a staff scientist at and the acting group leader for computational biosciences at Lawrence Berkeley National Laboratory. He's also an assistant adjunct professor at Helen Wills Neuroscience Institute and the Redwood Center for Theoretical Neuroscience at UC Berkeley, and also at the Kavli Institute for Fundamental Neuroscience at UC San Francisco. He is also the PI of the Neural Systems and Data Science Lab at Lawrence Berkeley at UCB, where he leads an interdisciplinary team that focuses on understanding how distributed neural circuits give rise to coordinated behaviors and perception, using a two-pronged approach, conducting in vivo neuroscience experiments, predominantly using electrocortography in awake humans, focusing predominantly on high gamma band activity, as well as developing and applying data science tools. And I'm sure that most of you are familiar with his work, but it is worth saying that he has provided some of the most impressive and important insights into the neural mechanisms of human speech production, specifically focusing on the functional organization of the human sensory motor cortex for speech articulation, the dynamic structure of neural variability in the cortical representation of speech, and the application of deep networks to predict produced speech syllables from high gamma cortical surface electrical potentials. And for those of you that don't know, in addition, he has also done some and been involved in some really nice work looking at the neural encoding and integration and dynamics of songbird sensory motor circuitry, which we'll save for another time, I'm sure. So without further ado, I will get out of the way and pass it over to Kristofer.
Well, thank you for that wonderful introduction, Vincent, and thank you for Mark and Vincent for the introduction. And hello, everyone. I'd like to thank you for the opportunity to talk to you about some of the work I did to understand how cortical networks are functionally organized and dynamically coordinated to produce human speech. This is work I did primarily as a postdoc with Eddie Chain. Now, I'd like to make the following disclaimers. I've been out of the field for a little while now, so if I make outdated claims, please don't hold it against me. Indeed, my own lab has been up and going for about 5 years now, and at the end of this talk, I'll briefly mention what we do. Also, for those of you who haven't figured it out yet, I stutter, and I'm happy to answer any questions about that as well. So we'll just get started. So here's a brief outline of some things to come. So we're first going to go over some work that I did to decipher the functional organization and dynamics of speech motor control. From there, we're going to transition into a detailed understanding of how the cortex controls vowel acoustics and co-articulation. And then we'll end with a couple of slides of what my current lab does these days. So as you all know very well, the ability to communicate through spoken language depends critically on the uniquely human capacity to produce the large corpus of sounds that compose a given language. Here is an example of one such speech sound, the consonant vowel syllable su, represented here as a spectrogram which displays the power as a function of frequency and time. The generation of speech sounds requires the coordinated control of the lips, jaw, tongue, and larynx to appropriately filter air against source at the larynx. Together, these articulators constitute a redundant plant with many degrees of freedom that needs to be controlled in rapid sequences. The coordination required of speech production is most apparent when watching the articulators in action during speech. So in this video, you're going to see data from a system we assembled to simultaneously record movement of the larynx using electroglottiography, the tongue captured using ultrasound, and the lips and jaw using video tracking.
Food.
Right, you get the basic idea. Now, the coordination required of behavior implies a similar degree of coordination in the brain. However, little is known about how this coordination arises in the cortex. Indeed, much of what we know about the cortical control of speech comes from electrical stimulation in neurosurgical patients. In this video, you're going to hear the subject counting while the surgeon stimulates different parts of the left hemisphere. This is a site in premotor cortex, and this is a site in the primary sensory-motor cortex.
24, 25, 20, 26, 27, 28, 29, 30, 31, 32, 33.
Such stimulation experiments have revealed that the cortical control of speech is hierarchically organized, with stimulation of higher order sites resulting, such as the frontal areas, resulting in speech arrest, while stimulation of the primary sensory motor cortex, which has direct projections to and from the face and vocal tract, results in twitches of individual orofacial organs. Furthermore, electrical stimulation has shown that the organs of the face and vocal tract are somatopically mapped onto the cortical surface, implying an orderly arrangement of speech body part representations. However, it is important to note that stimulation has never evoked a meaningful speech sound. To get a better understanding of the cortical control of speech, this research utilized electrocorticography, which places electrodes directly on the surface of the human brain. Here is a depiction of the electrode locations from a state-of-the-art 256-channel ECOG grid on the left lateral surface of a human. These grids are implanted as part of the clinical workup for epilepsy surgery and cover many parts of the brain involved in speech production and perception, such as the inferior frontal gyrus for broca's area, as well as the primary sensory-motor areas and Verticus area back here. Now here is brain activity during a speech production task, in this case, a simple consonant vowel syllable. Note the broad spatial patterns over this entire large area of the cortex and the very fast timescales. This underscores the necessity of these types of recording tools to understand complex behaviors such as speech. To understand how the human brain produces speech, we took a bottom-up approach and had neurosurgical patients read aloud consonant vowel syllables comprised of 19 consonants followed by one of 3 vowels. For example, we have ba, re, and su. These CVs span the range of American English sounds and are commonly found in many of the world's languages. Now, one behavioral note is that we aligned all our behavioral and physiological data to the acoustic onset of the consonant vowel transition. So most of the time when it's time point 0, that could be the consonant vowel transition. Okay, so our basic approach is to use the variety of movements required to generate different phonemes to understand how the brain produces speech. For example, the sound g is generated by constriction of the back tongue against the roof of the mouth. In contrast, the sound da is generated by constriction of the coronal tongue or the front of the tongue to the roof of the mouth. Finally, the sound B is generated by constriction of the upper and lower lips together. We will utilize the variability in articulator engagement across consonants to ask what is the organization of articulated representations in speech sensory-motor cortex. We used high-density electrocorticography to record neural activity from many sites in the ventral 1/3 of the U-shaped sensory-motor cortex, which comprises the precentral and postcentral gyri, as well as the gyral area at the ventral end of the central sulcus that connects the two, known as the subcentral gyrus. Let's first examine the activity of this electrode during the production of Ga. At each electrode, we extract the high gamma amplitude, which reflects deep layer multi-unit spiking and has high spatial and temporal resolution. For example, here we plot the mean and standard error measure of the high gamma z-score as a function of time, derived from multiple single trials during the production of the syllable Ga. Here, we see that elevated activity begins approximately 300 milliseconds before the onset of the consonant vowel transition and continues into the vowel phase. This temporal profile highlights or emphasizes the motor properties of the recorded activity. Now, let's expand our visual examination to include the activity of a few electrodes during the production of Ba, Da, and Ga, the consonants of which engage different articulators. Electrodes are going to be ordered and color-coded from red to black, with increasing distance from the sylvian fissure, which is this landmark right here. So again, here we're plotting the high gamma activity versus time from a dorsally located electrode. This most dorsal electrode is selectively active during the production of the labial consonant B. In contrast, during the production of the coronal tongue consonant D, we observed elevated activity in the more centrally located electrodes. Furthermore, during the production of the dorsal tongue consonant G, we observed the most elevated activity in this more ventrally located electrode. Finally, across these different consonants, we see that this most ventral electrode is consistently active during the production of the vowel A, suggesting a representation of the larynx. Together, these data suggest a dorsal ventral sequence of articulators. Now, to get at this quantitatively, we first approximated the articulatory requirements of the phonemes in our dataset with binary articulatory state variables. For B, the articulatory state vector indicates that the production of ba requires engagement of the lips, larynx, and jaw. In contrast, production of the consonant s requires the engagement of the tongue and jaw. So again, to understand the spatial distribution of articular representations, we are going to leverage the variability of articular engagement across consonants. Specifically, we model the high gamma activity recorded from single electrodes as an optimal weighted sum of the articulatory state variables using a general linear model. Here, we're plotting the spatial distribution of optimal weights for the lips averaged across 3 subjects on the Cartesian plane formed, on the Cartesian plane defined by the anterior-posterior distance from the central sulcus and the dorsal ventral distance from the sylvian fissure. Hotter colors indicate larger linear weightings and therefore stronger representations. We found that the representation of the lips was well localized to the dorsal aspect of BSMC. Here. Indeed, we observed that the lips, jaw, tongue, and larynx exhibited representations that were spatially distributed across the entire ventral sensory-motor cortex. When we looked at the spatial distribution of the largest articulator weightings, we found that the speech articulators were somatotically mapped onto the cortical surface. Here, each colored letter corresponds to the measured location of a strong articulator representation, and the colored pixels are areas classified by a k-nearest neighbor algorithm. In particular, we observed a dorsal-ventral sequence of articular representations going from larynx to tongue to jaw to lips and then back up to larynx. This speech-derived somatotopy, which is the first functional articulator map derived from speaking humans, is strikingly similar to the oral laryngeal homunculus described by Penfield through electrical stimulation of this same area of the brain in non-speaking humans. However, do note that we found an additional dorsal laryngeal area not described by Penfield. This dorsal laryngeal area is not found in non-human primates, and so may be a cortical specialization for speech production in humans. Subsequent work in the Chang lab has indeed identified this area as being specifically responsible for the control of pitch. Now, traditionally, the central sulcus has been conceived as a boundary between motor function in the precentral gyrus and sensory function in the postcentral gyrus. We therefore look for activation timing differences across the central sulcus that might differentiate the two. However, we found that the activations were similar across the pre and postcentral gyrus. This, along with other recent work, suggests broadly integrated motor and sensory function during behavior, but this remains poorly understood. Now, it is important to remember that the production of even the simplest of speech sounds, such as the consonant vowel syllables here, requires the precise and coordinated control of multiple speech articulators in rapid sequences. Furthermore, the somatotopic organization of speech articulated representations necessitates that multi-articular coordination gives rise to time-varying spatial patterns of activity across the surface of VSMC. That is, because the articulators are spatially distributed, the coordination of several articulators will manifest as spatial patterns of activity. To visualize these dynamic spatial patterns, I'm going to show you movies of the brain activity from one patient during the production of ba, da, and ga. Here, we've classified and color-coded individual electrodes according to the dominant articulated representation. The time-varying magnitude of the high gamma activity will be displayed as the opacity of the corresponding color. The spectrogram for an example syllable is displayed below, and this black dash line will progress through time. Just to remind you, time point 0 is the onset of the constant to vowel transition. So earlier times can be thought of as constant times, while latter times correspond to the vowel. During the production of ba, you see strong activation in the lip area, followed by jaw and tongue and larynx activation for the vowel. During the production of the coronal tongue consonant da, you'll see strong activation here in the tongue area in blue during the consonant phase that flows smoothly into the tongue, larynx, and jaw activation for the vowel. So this has, of course, been slowed down significantly so you can get a sense of the spatial patterns, not necessarily the dynamics. Now, I think you can appreciate that these videos show overlapping yet distinct spatial patterns of activity associated with these phonemes. We therefore wondered, what is the organization of phoneme representations that emerges from spatial patterns of sensory and motor cortical activity? Now, if we think of VSMC as a network and each electrode as a dimension, then the dynamic state of this network can be visualized by plotting the activity of electrodes against each other. For example, here we are plotting the activity of these three electrodes as a function of time during the production of ba, da, and ga. Now, to arrive at a quantitative understanding of the VSMC network dynamics underlying phoneme production, we used various dimensionality reduction techniques to derive a principled cortical state space that best describes the spatial structure of the network. For example, here we are plotting the spatial location of the syllables in the first 3 PCs at a constant time. We analyze the structure of the cortical state space with various clustering methods. For example, k-means clustering of the cortical state space identified these 3 groups of syllables. Indeed, we see that this blue cluster has syllables with dorsal tongue consonants such as k and g, while this gray cluster has coronal tongue consonants such as t and d, which you can't see here. I apologize for that. This suggests an organization based upon the engagement of the major speech articulators, dorsal tongue, coronal tongue, and labial. Now, many theories of motor control and speech control in particular have highlighted the utility of hierarchical organization of behavioral feature representations for action selection and control. We therefore tested for the existence of hierarchical organization in the cortical state space that had phonetically meaningful structure. Okay, so here's the dendrogram derived from the cortical state space at the constant time, which describes the organization of pairwise distances between syllable representations. Syllables linked by edges edges near the bottom have state space representations that are more similar than those linked by edges the farther up the dendrogram. We've colored the dendrogram to reflect the same clustering as before, with the corresponding syllables in light gray. Next, we're going to color code the syllables according to the articulatory engagement during consonant production. This clustering, which is defined purely based on the cortical data, organizes the consonants according to the major speech articulators, demonstrating the cortical state is primarily organized according to these dimensions. Indeed, we found that the cortical state space had phonetically meaningful hierarchical structure. For example, we found that the constriction location and the front tongue imparts secondary structure, while constriction degree imparts tertiary structure. We quantified the hierarchical organization of phonetic features by correlating the distances between syllable representations in the cortical state space with the distances predicted by different phonetic features. Here, we're applying the distribution of correlations across subjects and consonantal time points. We found that constriction location accounted for significantly more structure than constriction degree, which in turn accounted for more structure than the acoustics of the upcoming vowel features. Correspondingly, at a time appropriate for vowel control, we found the cortical state space organized the syllables according to the 3 vowels a, e, and u, and also exhibited some degree of hierarchical structure, such as the primary split based on lip rounding for u versus e and a. Across subjects and vowel times, we found the cortical state space was significantly more correlated with the vowel configuration than with features of the preceding consonant. Do note, however, that vowel features nonetheless imparted significant structure during the consonant times, right here, and the consonant features imparted statistically significant structure during the vowel times. These observations provide a hint of co-articulatory effects that we'll return to that issue in much more detail later in the talk. Now, the dramatic differences between the organization of syllables in the cortical state space via the consonant times and the vowel times led us to wonder, what are the dynamics of phonetic representations in VSMC? Now, the dynamics of neural populations have been shown to reflect important properties of the underlying neural network. To gain insights into the dynamic organization of VSMC during speech articulation, we first visualized the trajectories of individual syllables in the first 2 PCs through time. Here, we're applying the trajectories for the individual front tongue and dorsal tongue consonants transitioning to the vowel u. For example, these are the trajectories for the front tongue consonants tu and du, and these are the trajectories for the back tongue consonants gu and ku. Initially, the representations of these distinct consonant sets are intermingled and poorly separated. As time progresses, the different consonant sets diverge from one another while simultaneously exhibiting within cluster convergence. As these consonants transition to the common vowel oo, we see the trajectories converge to a common region and then diverge randomly, presumably as the articulators are returning to a neutral position. Across the different consonant sets, we observe that phonetic structure emerges through synchronous cross-cluster divergence and within-cluster convergence. Together, these divergent and convergent dynamics increase the separability of the different consonant sets during production. We observed analogous cortical dynamics underlying the production of the three vowels. Here, we plot the trajectories of the labial consonants, transitioning to a, e, and u. Similar results were observed and quantified for the other subjects, but I'm not going to get into that level of detail right now. Finally, the dynamics of the entire cortical state during the production of all CV syllables is summarized here. Now, there's a lot going on in this visualization, so I'll walk you through it. In this visualization, the center of each colored tube is located at the centroid of the corresponding phonetic cluster. Tube diameter corresponds to cluster density, and the color saturation corresponds to the time-varying correlation between the cortical state and phonetic features. This visualization reveals several interesting features of the sequential cortical dynamics. First, with all CVs on the same axes, we observe that consonants occupy a distinct region of the cortical state compared to vowels. This is despite the fact that consonants and vowels engage the same articulator sets. Secondly, the consonant to vowel sequence exhibits a periodic structure with further specifications for the consonant and vowel features. These specifications can be thought of as subspaces through which the cortical state must travel to produce the corresponding phoneme. All right, I'm going a little fast, that's okay. All right, so up to now, we have been examining the cortical control of speech through the filter of averages. That is, we have been examining the relationship between mean neural activity and coarse-grained descriptions of speech articulation. This has been very informative at revealing the high-level structure of the relationship between brain and speech. However, there are several questions that can only be addressed at a final resolution. So to move forward, we investigated how the structure of speech motor control of variations in production of the same utterances and the context dependent of this control. We focus on the vowels because the relationship between a specific acoustic features and articulator movements and position is more direct than for the consonants. Here, we are plotting single utterance formants and a pitch extracted from the vowels a, e, and u during the consonant vowel task I described previously. Below, we have the single utterance activity from one ECOG electrode during multiple productions of each vowel when preceded by the consonant g. While there is clearly large signal on single trials that bears some information to the identity of the vowel, it is not clear from visual examination of this raw data that there is further information about within vowel fluctuations. To mathematically examine the degree to which fluctuations in various acoustic features are present in these data, we are going to use the single trial ECOG data from all BSMC electrodes and predict the steady state vowel acoustics of single utterances extracted during this time window right here. In this scatter plot, each little letter corresponds to an actual F2, F1 ratio for the vowels a, e, and u. versus the optimal linear predictor of that ratio from the population ECOG data. Here, we train cross-validated linear decoders separately for each vowel to focus on the within-vowel fluctuations. Across 3 patients, we see that we are able to get decent predictive accuracy, but it's clearly not perfect. Also note that there are differences across the different vowels. Indeed, when we look across a variety of acoustic features, we see that we are always predicting the behavior from the ECOG much greater than expected by chance, where chance is here in black. But there is large heterogeneity across features and vowels. This raises the question of what is driving this variability. So, for example, for U, we generally have the largest predictive accuracy in the F2 and F2 relevant ratios, and in similar but somewhat more muted patterns across the different vowels. Now, so far we have been primarily treating consonants and vowels as non-interacting objects. However, at a behavioral level, there are interactions between the production of phonemes, a process known as co-articulation. Co-articulation comes in 2 main flavors: anticipatory co-articulation, in which the production of a given consonant or phoneme will vary slightly depending upon the upcoming phonemes, and perseverative co-articulation, in which the production of a phoneme, in this case a vowel, will vary slightly depending on the preceding phoneme, in this case a consonant. Co-articulation is the primary reason why speech production is not simply the concatenation of discrete tokens, but instead reflects a smooth trajectory through the sequence elements. Now, the origin and function of co-articulation is a major debate in modern linguistics research, as well as the study of other sequential behaviors. So we first looked for behavioral evidence of co-articulation in our data. So here we are plotting single utterance F2 F1 ratios as a function of time from the now aligned to the onset of the consonant to vowel transition. We colored each line or each utterance by the major articulator engaged by the preceding consonant. There is clear impact of the preceding consonant articulator on these acoustics. For example, the F2 F1 ratio was higher when preceded by dorsal consonants for U, such as duh, than when preceded by labial consonants such as buh. To quantify the impact of consonant articulators on vowel acoustics, we constructed a linear model that predicted variations in vowel acoustics from a binary representation of the consonant articulators. Here, we are plotting the cross-validated R-squared for predicting the F2 F1 ratio as a function of time, which demonstrated clear evidence for perseverative co-articulation at the times during which the vowel times that we use for decoding, demarcated here in gray. And this was true variously for the different vowels in a way that is in agreement with linguistic understanding. Now, interestingly, across acoustic features, the pattern of co-articulation, as quantified by the R-squared of the acoustics for the preceding consonant articulators, qualitatively matched the pattern of decoding accuracy. So, for example, you can see this here, higher here, and then similarly across different vowels. This suggests that co-articulation may have a signature in the cortical activity. For the next couple of slides, I'm going to focus on results for U, for its co-articulation effects were largest, but analogous results were observed for the other vowels. So to quantitatively examine if cortical activity controls co-articulation, we first examined how decoding accuracy changed when decoding the full acoustics versus the residual acoustics after where we have now removed the effect of co-articulation using the linear model I just mentioned. So for each point in time, we assessed how well how well we could decode either the full acoustics here in black and compare that to the decoding accuracy of the acoustic residuals in red after removing the effects of corticulation. Here, time is measured relative to the onset of the acoustic measurement, so that orange time points correspond to the correspond to the consonant, and the yellow time points correspond to the vowel, respectively. We found that at both vowel and consonant time points, removing the effects of core articulation reduced the ability to decode the acoustic feature. Indeed, during both vowel and consonant times, we observed that removing the effects of core articulation resulted in significant decreases in the ability to act in our decoding accuracy for exactly those acoustic features that exhibited the strongest effects of core articulation. This demonstrates that the cortical activity is controlling both anticipatory and perseverative core articulation. Now, while the previous analyses demonstrates the existence of cortical control of anticipatory and perseverative core articulation, it leaves unspecified the structure of that control. In particular, arguments from optimal control theory suggest that the state space for one phoneme should be biased towards the state space occupied by the adjacent phoneme. To examine how the cortical control of single phonemes were affected by surrounding phonemes, we again utilize a cortical state space framework, this time using linear discriminants analysis of specific syllable contrasts to define the axes. Keeping our focus on U for illustrative purposes, here we are plotting the mean plus or minus standard error state space trajectories for the vowel U transitioning from the consonants B, D, and G. Again, derived from single trial data. During the vowel, we see that the trajectories through a large portion of the vowel U are biased towards the state space, the subspaces of the preceding consonants. Thus, there is a perseverative bias in the cortical activity for the vowel U that reflects the location in cortical state space of the preceding consonant. Analogously, and more interestingly, for the single consonant G, we see that the trajectories start to diverge towards the upcoming vowels during the consonant times, biasing the trajectories through the consonant subspace toward the subspaces for the upcoming vowels. Thus, this is an anticipatory bias. Now, although I'm only showing examples, these phenomena were robustly observed and quantified across several syllable contrasts and 3 subjects. All right, so the results I just described were focused on the vowels, and we only had acoustic measurements during the consonant to vowel task. And the correspondence between articulators' acoustics is most pronounced for vowel formants. To enable single trial analysis of speech articulator kinematics, we created an articulatory tracking system compatible with data collection from ECOG patients. Here is the colored face of one of the patients, which makes extracting the lips, jaw, and nose very easy, even in the kind of poor and variable lighting conditions that are present in the hospital monitoring room. Indeed, once I start, as you can see from this video, we can extract the various kinematic properties of the lips and jaw very easily, such as the vertical velocity. Now, we collected that data during a task specifically focused on understanding the temporal extent of corticulation, in which we had the subjects produce oo sounds while varying the sequence of the preceding consonants that were neutral for lip rounding. So this really enabled this task and directly enabled us to monitor how far in advance does the brain and behavior of lip rounding anticipate the upcoming vowel oo. Now I kind of ran out of time during my postdoc. Oops, sorry about that. I ran out of time during my postdoc to examine that, but preliminary results demonstrated that we were indeed able to predict such kinematic features from ecog signals with high accuracy. In this case, lip aperture. So this is the measured lip aperture in some pixel units versus the predicted value, in this case, just using a Kalman filter. But using this experimental paradigm with a graduate student in the Chang lab, we have since gone on and done a detailed study of the direct relationship between cortical activity and oral facial articular kinematics. You can find that work described here. All right, so I'm a little bit ahead of schedule, but that leaves us a lot of time for questions. So in summary, my work has defined the anatomical map of individual articulated representations of cortex, as well as revealed the cortical dynamics and hierarchical latent structure of speech motor control. Finally, I didn't have time to talk about this, but we have also closed the loop by using the IBM True North neuromorphic chip as a low-power, high-performance computing platform for brain-machine interfaces. The work I described today can be found in these papers, as well as other work that I did in speech control topics. Okay, now in many ways that was work from my previous life. So fundamentally, I'm just a neuroscientist. Well, turns out a problem. And as such, the work in my, and I'm also not a neurosurgeon, so studying the humans is challenging. Sorry. As such, the work in my own lab aims to carry forward the central dogma in biology that structure defines function, which gives rise to phenotypes. Our goal is to derive a quantitative and predictive understanding of how the biophysics of widely distributed coordinated neural activity that ultimately produce complex behaviors and perceptions. One such behavior is rat reaching, where we have developed a pneumatic robot that automatically positions a reward delivering handle in 3D, and we can track the position of multiple points of both arms during this task. This will allow us to, for example, understand how the spatial dimensionality of the task modulates interareal communication between premotor cortex and primary motor cortex. Indeed, we use a variety of neurophysiology techniques to measure brain activity, both during behavior such as rat reaching, as well as during perception. For example, here is a visualization of the activity of an 8x16 grid of electrodes placed on the rat auditory cortex in response to a Tippett sentence. Here, each electrode is colored according to the auditory frequency that gave the strongest response, and the opacity corresponds to the magnitude of the neural activity. Note that time in both of these videos is greatly slowed down. And in particular, for the neural one, it really emphasizes the rapid dynamics of the highly distributed neural response patterns evoked by this complex stimulus. Finally, let me just pause these. Finally, to understand the biophysical mechanisms of our measured neural activity, we use large-scale biophysical simulations informed by experimental data. Here, we're visualizing the membrane potential of 30,000 neurons that comprise a cortical column, with each neuron having approximately 1,000 segments. Now, one thing I think you can all appreciate from these videos is that across all these spatial temporal scales, brains and behavior are highly dynamic. So a major focus of my lab's analytic work is to derive methods and frameworks for understanding dynamic data. So generally speaking, brain dynamics generate diverse sensations, behaviors, and cognition through the flexible engagement of networks of many neurons and brain areas. Now, of course, we have to extract functional networks and latent dynamics from recorded data. Analytically, dimensionality reduction and latent state methods extract subspaces that provide compact descriptions of neural populations, such as I demonstrated in my talk. But they do not address the role of individual observational components, whether those be ECOG electrodes or individual neurons. Concomitantly, network inference methods attempt to directly describe the coupling among constituent components, but suffer from many statistical shortcomings from a methodological standpoint. This is why we've gone back to fundamentals and created two new methods for inferring functional coupling networks with our UOI vector regressive methods, as well as latent dynamical subspaces with our dynamical components analyses. And here are the references for those methodological in NeurIPS. Now, while I won't show any results for this, any results for this one, this work has shown that the motor cortex controlling behaviors such as speech and arm reaching is accomplished from a sparse subset of signals interacting in sparsely coupled networks. And finally, we are developing new mathematical theory to analytically link dynamic subspaces to functional networks are inferred from data. All right, with that, I'd like to make sure that people recognize that these are the people in my group who do all the actual work. I just talk, raise money, and write papers. And with that, I'd like to thank you for listening, and I'll happily take and potentially punt on any questions you may have. Thank you.
Thank you. And punting is allowed, so.
I appreciate that.
So there are a few questions. Let's start with, are the patterns of tongue activity, this is from M. Smith, are the patterns of tongue cortical activity after the BA information about the tongue returning to some sort of neutral position prior to the next utterance?
That is presumably what is going on, but we do not have direct articulatory monitoring during that, so we cannot say for certain. It is also the case that we observed two distinct types of vowel production in this task. One where the subjects held the acoustics and hence the articulatory positions throughout the entire vowel and then ended it short, kind of abruptly, and others where they had this kind of like trailing nature to it, where they were kind of engaging the larynx and therefore expiring, but clearly bringing the articulators back to neutral position during this task. And that's kind of why we focused on that initial steady state version, so that we wouldn't have to because we don't have good ability to distinguish between those distinct end vowel production states.
Yeah. Okay. So I would I would encourage people, because we have about 8 questions, that you can vote on the questions. So otherwise, I'm just going to go in order. The next question is also from M. Smith. Are the patterns of already where'd it go? Oh, here we go. Sorry. What do you predict for, or do you predict anything for cortical activity in speech errors, where there unintended syllables are produced? Have you seen any of that? I would imagine you have some speech errors.
Well, so several things. One is that the task that was produced was a very simple consonant vowel syllable reading, and we looked at what they actually said, not what they were supposed to say. And my intuition says that in this part of the brain, the ventral cortex, which is directly responsible for the control of the oral facial articulators, we would primarily see engagement that are related to what was actually said, not to say what was intended to be said. Now, we are also able to monitor the inferior frontal gyrus, i.e. Broca's area, during that time as well. And I think it would be fascinating to look at are there communication kind of issues between Broca's area and BSMC that correspond to errors in production as what was being kind of articulated. But that gets tricky very, very fast, and more methods would be required to look at that.
Yeah, probably more data too.
Yeah,
so from Bernd Kroger, why are you? He would like to know if there's some justification for looking at syllable organization in primary motor and sensory cortices and not in higher level cortex, like premotor cortex?
Sure. So a couple of things. One is that premotor cortex is not an anatomically defined thing in the humans, right? So it's a functional separation of the precentral gyrus, and we do not have access to that level of anatomical description in our data. We picked that part of the brain because it was very accessible, was poorly understood when we looked at it, and it gave kind of very robust, large magnitude signals that were kind of easily analyzed. You know, with the brain, things are complicated enough, so it's often easiest to start with things closer to the periphery, understand that in great detail, and then work our way up the cortical hierarchy, because things will always kind of depend on lower levels. So it's just a general brain approach thing.
Yeah. Okay. Next question from Fabian Tomaschek. How often did the speakers have to articulate each syllable? If they were produced multiple times, do you analyze the activation patterns as a function of repetition, like first half of the experiment versus second half? And if yes, what did it look like? And if no, what would you predict?
So each syllable was produced on the order of about 40 to 45 times in a pseudo-random order. We did not look for sequential effects. That's very hard to do for a variety of technical reasons, just because the recordings in the hospital are done in different recording blocks. So they would get through order 10 utterances per syllable in one block, and we had to come back the next day. So there's just constraints about doing this kind of work in neurosurgical patients that make it a challenge. And we did not look at serial repetition because the syllables were not repeated serially. They were repeated in a pseudo-random order.
Yeah, okay. So I'll take Pascal's question first. Do you have any way to statistically assess whether anticipatory and retentive co-articulation result in different or similar amounts of change in cortical activity?
Yeah, that's a great question. I think the way that I would assess that is, again, by looking at our decoding accuracy during the regular constant production and the residuals after when we have removed the effects of co-articulation. And there we do see that the magnitude of decoding drop is largest during the vowel times, or it's larger during the vowel times corresponding to the perseverative co-articulation than during the anticipatory times. I think that also just makes sense from the magnitude of the articulatory effects that are observed in anticipatory versus perseverative co-articulation as well.
Yeah, okay. Question from Matthew Fatak, who thanks you for your talk. In the Nature paper about somatotopic organization, vowels, consonants end up in different parts of reduced dimensionality space. Might this merely be because the articulators recruited for production of d, g, and s, and so on aren't exactly the same as those recruited for the vowels? For instance, in the last tubes figure, it'd be much more conclusive to see, for instance, the semivowels, E and W, and the vowels E and U embedded differently in the reduced dimensionality space, since they recruit maximally similar articulators, but differ by whether they're consonant or vowel.
Yeah, I'm finding a little bit hard to parsing what the question is, but it is true that we can only speak to the data, the structure of the data that was revealed by the task at hand. It is clearly the case that having vowels that are more consonant-like would likely reduce the difference between the consonant and vowel subspaces. That's a bit hard to assess in our data set. It's also the case, however, that please do note that that there were distinct electrodes that were active during the vowels that were not very much active during the consonants, and these are particularly the laryngeal electrodes. Right. And so as one would expect, so as long as that laryngeal electrode is highly active and much more active during the bowel phase, which I think it will be both during the increased amplitude of the bowels, I think that we would still nonetheless see distinct cortical state spaces for the bowels because they're occupying just like a different source of variance is very important for the productions.
Yeah. So let me ask David's question, David Ostry. What do you pick up with ECOG electrodes? You get layer 4, 5 information, or are you limited to cortical-cortical interactions in layers 2 and 3?
Yeah. So David, that's a great question. And it's not, unfortunately, it's not a well-defined one. And I say that for the following reason, right? And an ECOG signal is only defined once one defines the electrode itself. And because ECOG electrodes come in a variety of shapes and sizes, it's not quite a well-defined thing. So I can't speak to that in humans, but in rats, I have an answer, right? So there we've taken ECOG, shrunk it down to micro size, where each electrode is now 30 micrometers. We can do that because we can electroplate the electrodes to have low impedance. We can't do that in the humans just for safety reasons. And that means that each electrode is on the order of the diameter of a cortical mini column. And with that, and so we put that on the rat brain, and there we have been able to demonstrate that at least the high gamma activity is localized to single cortical columns. What I mean by that is that you can look across electrodes at 200 micrometer pitch and see different response patterns in the rat primary artery cortex. Now, we are using our biophysical simulations, which are very accurate forward models, as well as simultaneous laminar polytrode and ECOG recordings, where we are recording single units from across our cortical column, and we can now build statistical forward and statistical inverse models of all these things. Now, long story short, is that our current understanding of it from all those experiments is really suggesting that the high gamma signal at the surface is localizable to a single cortical column and primarily reflects multi-unit firing rate of deep cortical neurons. And this is because there are just many more neurons in the deep layers. So right, there's 3 main factors that contribute to signal propagation: distance, correlation amongst the sources, and number of sources. And it just turns out that there's a shit ton of neurons in layer 5 and in layer 6, and that tends to just dominate. At least our current understanding is that those effects dominate the distant effects.
Yeah. So there are a few more questions, and I think we have the time.
Yeah.
I want to throw one in because I know you've done this work, and just I'd like to hear your response. So you spend a lot of time looking at high gamma activity, but I know you've also looked at other activity, and there tends to be a focus more primarily, I think, because people are not in the ECOG literature, but in EEG literature, to focus on beta, for example, and lower frequencies too. But I would imagine the lower frequencies, you're not going to see a whole lot in sensory motor cortex because you're not looking at long range connections. But how does beta activity fit in with high gamma?
Yeah, so Vincent, you clearly read that PLOS Comp Bio paper, so as you know, it's not going to be a short answer, but I'll do my best. So you could just organize my thoughts about this one. All right. Cohn, Nathan Cohn, back in late 90s, early 2000s, I forget what the exact date was, kind of described this effect of beta, or what he called beta desynchronization during behavior in BSMC. And this was observed as a decrease in beta power, which is not necessarily the same as desynchronization, right? Because desynchronization is a comment about time. What he observed is a decrease in power. So that's a bit of a naming thing, but fine. And he observed it at the large electrodes with the coarse spacing. And it was predominantly looked at when aggregated across all the speech production tasks, across all the electrodes. Now, when we looked so we found that same basic effect, right? So when we looked at the different cosmological symbols across all the electrodes, you see this basic kind of reduction of beta power and a decorrelation of high gamma and beta when you kind of aggregate across all the electrodes. However, it's important to note, right, that individual speech sounds engage sparse subsets of electrodes, and because there is a tight correspondence between electrodes, between articulators and electrodes in VHMC, that dictates that there is a correspondingly sparse subset of electrodes that are active on any individual speech sound. So when you aggregate results across all speech sounds, or across all electrodes for a single speech sound, you are primarily going to be picking up on electrodes that are actively disengaged during during the production of any given speech sounds. So what we did, and this is not a short answer, we took all our electrodes, all our speech sounds, and we just segregated them according to those electrodes that have a large high gamma response during the speech sounds and those that do not, right? It doesn't matter what threshold we use, just that's how we split the data. You then can sort that and kind of look at how the beta activity at the active versus the inactive electrodes is related to high gamma, as well as the overall power. And there we see a very different story, right? First, that we see this decrease in beta power only at the electrodes that are not actively engaged in speech production. Indeed, when the electrode is engaged in speech production, as indicated by having a large high gamma response, we see an increase in beta power and a non-trivial correlation between the beta power waveform and the high gamma waveform. Now, and this was not just some like arbitrary thing, right? It was very spectrally specific to right around like the high beta circa like 23 hertz types of thing. So interestingly, if you look at the active electrodes, at the correlation as a function of frequency, where now this is frequency of the low frequency signal with and the correlation of that low frequency signal with the high gamma band power, we see that it starts off high with the low frequencies, has this big dip, and then this really like spectrally specific peak at 23 hertz that then dives back down and increases back up to the gamma. So there's a lot of phenomenology, but it highlights two important points, right? One is that we need to be very careful about interpreting signals when we are aggregating across large masses of electrodes that may or may not be engaged in the task at hand. And it does bring up a bit of a question about some of the theories that people have about beta as being, and its role in certain motor coordination, as well as theta for that matter. You know, one thing that I would speculate on, and this is just pure speculation, although there is some data to suggest it, is that we see that, so we think that beta is indeed reflecting a coordinating activity in the brain, but it is increased beta activity at those electrodes that are actively engaged in the task, that are being coordinated, where we see kind of increased theta modulation or coupling at the electrodes that are not being actively engaged in a task. So it's like we have these two bidirectional pedals about whether or not we want to engage or we want to coordinate electrodes that are actively engaged in task versus kind of disengaged and keep the other parts of the brain just quiet. And that's being coordinated by theta. So I kind of just made that up, but it's supported by the results that we have.
Yeah, well, that was good. That was good. And kind of related, I think you kind of answered this already, but Doug Shane had a question. Is the assumption that cortical gamma activity reflects mostly excitatory output to the muscles or contraction, or could it be indicating inhibition of movement as well?
So two things. One is that we need to be careful about distinguishing between gamma and high gamma. These are not the same things. They're not the same things statistically in our signals, not the same things biophysically. So we need to be more, I mean, my big gripe with the ECOG community writ large is that they've been kind of loosey-goosey with how they kind of define things, and some of the theories that they put out there are not really well quantitatively defined. So that's my kind of little spiel, deal with that. So Right. High gamma, which only thing we're going to talk about, primarily reflects excitatory neurons. And this is because they tend to have larger somas and dendrites, creating large dipoles. These large dipoles create large action potentials. And so the other term that plays into the degree to which any given source contributes to a aggregated signal is the amplitude of that signal. And action potentials generated by excitatory neurons are just larger than that by inhibitory neurons. At least this is what our initial results are showing, and I presume that things will be different. Now, this is why gamma is also a different thing, whereas gamma, I think, is primarily, at least my current understanding is that gamma, which is order 50 hertz or so, is primarily produced by parvalbumin positive inhibitory interneurons, and that's been demonstrated through a variety of both optogenetic and electrophysiological monitoring techniques.
Yeah. Okay. It's a question from Jen Pardo. Did you examine whether cortical representations of syllables clustered at all according to the speakers?
I don't know how to address that question because we can only look at within speaker organization, and every speaker said their own syllables. There is no auditory part of this task. And so it's just, they were our list reading. So I don't think there's a way for us to meaningfully get at that specific question.
Okay.
I mean, if I could expand on that a little bit, so I could imagine, right, that if one had like, speakers from different languages, then because different languages have, like Mandarin, for example, has a entirely different class of sounds they produce, then I would imagine that you would see within language clustering more than across language clustering. But I think that the organization of the articulators would be, that's genetically encoded, right? And just worked out through development, through talking and eating. And so I think that unless there's something very seriously wrong with the brain of a person, that somatopic organization will be the truth of how the brain is organized within some margins of error, of course.
Wow. So another question from Carrie Roundtree, who said, "This is so interesting. Thanks for a great talk. Appreciate the tight temporal link to the acoustic signal in your visuals in the first half of your presentation. If you're familiar with FNIRS, what do you think the application of this work could be to researchers who could use FNIRS to look at the same or more functional speech tasks?"
Yeah, so I'm only vaguely familiar with FNIRS through a collaboration I have with the Gates Foundation. Let me think about this for a moment. Right. So my understanding, right, that FNIRS uses IR to go through the skull, and the skull gets thicker as we get old. My skull is very, very thick, so I'm very old these days. And so it's not clear to me the degree to which the signal recorded by FNIRS has the sufficient spatial and temporal resolution to kind of study the things that we were investigating here. And this really required not even kind of like regular ECOG data, but like these high density ECOG arrays, which had 156 channels versus 64, smaller electrode contacts and tighter inter-electrode spacings. Things happen very, very quickly, order 100 milliseconds or so, and the ECOG, the high gamma signal in ECOG itself has a temporal resolution of order 10 milliseconds. And so it's just not clear to me if FNIRS has sufficient spatial temporal resolution and density of coverage, to be specific, over the parts of the brain that are relevant for speech production and perception. Yeah, so I just don't have enough technical detail about that. I apologize.
Yeah. Well, certainly not at the resolution that you're looking at, for sure.
Yeah.
Question from, Xiaoyan Zhang. Is the electrode spatial resolution fine enough to differentiate between individual laryngeal muscles, for example, for vocal control?
Two things. One is almost certainly not, but it's also not entirely clear the degree to which there is a myography in the brain, right? So here, this is important. So there's a somatotopy, which means a layout of the gross anatomical locations of the body parts in the cortical surface. What you're asking is, and this has been looked at both anatomically, electrophysiologically, is a myography, right? Which says there is a mapping of locations of individual muscles and those fibers onto the brain. Now, again, in humans, I do not know of any data that suggests that exists. In monkeys, it's not very good, and this is all done by anatomical tracings, which suggests that there is not a myography. Electrical stimulation in both humans and non-human primates suggests that you can't observe it in that way. However, in mice, those damn mice, so what they can do is use optogenetics to infect neurons to retrogradely label neurons in layer 5 that project to specific subgroups. I think this was like bicep versus tricep. I could be making that up or something like that. And there, they found very precise innervation patterns between specific neuronal populations in layer 5 that project to different parts of some muscle in the arm of the mice. And they demonstrated that through kind of precise optogenetic stimulation. But whether or not that exists in primates is, as far as I know, unknown. It's a good question, though.
Okay, we still have a couple more. From Pierre Beraduc. Did you record an M1, S1 during inner speech? And if yes, did that correlate to overt speech? So inner speech is like motor imagery, yeah? Okay.
No, we did not do that. Inner speech is, that's some hard stuff. So several things. Writ large, non-vocalized speech has a very weak neural signature. And this is true in BSMC, in Broca's area. Just the magnitude of the signal is greatly reduced. Now, of course, that brings with it a whole host of other just analytic issues of interpreting whatever that is going on. I can't recall. So I certainly not do that. I think that people in Eddie's lab have subsequently tried to look at that. It's hard. I don't recall of any strong results that kind of speak to that. But again, that's just not clear to me if they would see it, given that there's no actual engagement of the muscles that produce speech, which is exactly what the neurons in the VSMC are actually driving.
Yeah. Okay. Another question from David Ostry, who says one more question. There may be another one too. There's lots of activity in somatosensory cortex. What's happening here? What is the timing of this activity? Are you seeing motor efferents from somatosensory cortex or sensory receptive fields in M1?
So I get this a lot, and I think that this is due to the kind of canonical and classic understanding of a parcellation of function across M1 and S1. During behavior, right, and at the level we are recording at, it is well understood that these two parts of the brain operate as a single integrated whole. So it is true that one part is projecting and one is receiving, but they are so tightly integrated at such at short time scales that it's probably most accurate to think of these things as one tightly integrated network than as distinct circuits during the actual behavior. When we've looked at this, we have found no evidence of timing differences across the central sulcus. When people have done things in mice, they also kind of find very short latency timing differences between them that is again indicative of a tightly integrated circuit during behavior. So I cannot speak to those issues. We have tried to look at sensory receptive fields in ECOG before. So here we were using Von Frey hairs, and we were kind of like, touching the face. That's kind of boring. Touching the mouth is kind of hard because you gotta kind of get in there. We did it in a couple of subjects. It wasn't a particularly well or carefully controlled set of experiments, and so I don't think we even looked at the data particularly well. So I don't know.
Yeah.
Although it is the case that during stimulation, right, You also here do see this big distinction between pre-central and post-central, whereas if you stimulate post-central, you typically get like tinglings, and if you stimulate pre-central, you typically get like little like fluctuate, like nonsensical fluctuations of the articulator.
Yeah. Well, so I think those are all the questions, but I still have one or two, if you don't mind.
This is probably the last time I'll ever talk about speech, so have at it.
So I was trying to remember, do you ever record on the right hemisphere?
We do, but but these were all on speech in the speech dominant hemisphere.
Yeah.
So we have not looked at that explicitly. There was a paper from what's his name? Some guy at NYU, whose name I'm misremembering now, that says that at least at the level of BSMC, like primary sensory motor control, there isn't much lateralization. And we know that from various lesion studies as well. I mean, this is the part of the brain that bilaterally innervates the face and vocal tract. So we don't see much differences, or it's not. I would be surprised if we saw much differences at the level at which we're investigating things between the dominant and non-dominant hemispheres. But that's just speculation.
Yeah. So the reason I ask is that there was a study a number of years ago, a functional imaging study where they did kind of this orthogonal manipulation of different syllables to look at the representation of the larynx, the lips, and the tongue, and maybe the jaw too. And they found that in the left hemisphere, there was well segregated regions, kind of like what you've shown, but in the right hemisphere, it wasn't there, which is kind of interesting. I mean, on the one hand, we know that speech is bilaterally controlled, but are they the same, or does one complement the other, or something like that? But anyhow, if there's no data, it's hard to know.
Yeah. I don't know that specific study. At least I can't raise it to my level of consciousness right now. I will note that the specific task and experiment will change results, right? So we looked during kind of relatively freely speaking conditions. If one is manipulating the vocal tract or manipulating things in various ways, it's kind of all bets are off at that point, because now you're looking at like sensory motor integration and control, which is a slightly different thing than kind of like the feed-forward control signals that I think of that we were kind of mostly studying. Does that make sense?
Well, yeah, it does. But it leads into my last question, and that is that a lot of the stuff that you looked at was essentially repetition, if I remember correctly, right? So to some extent, while you can think of it as being feed forward, you can also think of it as being primed, right? Because the auditory ultimately impacts, we know at least from functional imaging studies, that there is some kind of effect on the motor cortex of things that you're hearing, whether it's from externally or your own feedback. And so I just wonder if part of what you're seeing is a priming of the motor cortex based on what they're hearing, and the way to look at that would be to have them produce speech where they're doing it kind of free form, right? And I know you talked about this, but in one of your papers, but I don't know if you've actually done it.
Right. So a couple of things. So just so we're all kind of level set. This was a reading task. So the subjects basically had like a list of consonant vowel syllables that they read through at their own pace, right? Which was good and bad, because it provides some experimental caveats that we were able to kind of deal with. Now, it is true that they were hearing themselves while they were vocalizing. During the consonant time, at least for like the stop consonants, there's basically no acoustics going on there at all, right? So that is, for those consonants in particular, I feel very confident saying that is primarily a feed forward signal. The vowels, all bets are off, because by that time the consonant acoustics are coming back in, and the vowels are sufficiently prolonged that they're also getting feedback, auditory feedback from their own vowels during that production. And indeed, if you remember way back when I showed you the video of the entire ECOG grid, there was clear activation in the VSMC, but also at a lag, there was activation in the superior temporal gyrus, which is presumably where the auditory feedback is being processed to go back into that larger loop. So, the only thing I can say is that we did as best as we could to segregate out those effects. In anything that is sequential like this, it's not possible. As you know, the consonants themselves cannot really be vocalized without being followed by some consonants, particularly like the stop consonants, because there's no, right? But these are all great questions. It's to be determined. I will note that we did see a different pattern of correlations between high gamma and lower frequencies in the superior temporal gyrus than we saw in the VSMC. And it was also different in the in Broca's area, in the IFG as well. So there's some potentially interesting things to dig into in these types of data sets. But I ran out of time because I had to go on with my life.
Well, too bad for us, but good for you. So I think we've exhausted the questions and we exhausted you.
Well,
and,
I do want to say I thank you all for this wonderful opportunity to talk. It was something like a farewell to the field. I had a really wonderful time interacting with many of you. Speech is a phenomenal behavior to study, and I do find it interesting that I started off studying birdsong. I then studied human speech, and then I have this thing to deal with, which is kind of a pain in the ass. But no one ever asks me about it. So it's kind of remarkable that I give talks about speech production. I stutter, but no one ever asks me about it. So that's probably because you are all far too more polite than I am. But if you're at all interested in this, I'm just now starting to kind of, stuttering is weird. I'm a weird individual, so I'm just now starting to think about kind of engaging with that stuttering community. And I'd be happy to kind of talk to anybody about that. Probably offline is the best place.
Yeah.
Right.
Okay.
Thank you, everyone.
Well,
this was fun.
Yeah.
Bye.
And